this work started early 2000. And so I've been working on this problem for 2.5 decades. And I realized very early on that it's very important to work together on a project like this. It's very difficult for one research group to solve all these problems because it is really complex. But the aim, of course, was at that time and has been to be able to manufacture these insulin-producing beta cells and to transplant them into patients with type 1 diabetes and to test whether they actually would help the patients to regulate their sugar levels in a much more precise way than what is currently available with the sensors and pumps. And we can come back to that later. That's actually quite a high bar to overcome. But you have to, since these cells normally handle this in our body, it's really hard to see how something could outcompete these cells if you could put them back into the patients. So I've been leading 2 EU programs where I have gathered really fantastic scientists all around Europe to work on these problems. The first consortium was called HUMAN, and it was more working on more basic questions about, how do we actually make these beta cells from stem cells in order, basically to develop differentiation protocols to make these cells in tissue culture dishes. Was also to try to understand if we can make these cells, how are we going to ensure that we can make enough cells if it turns out that these cells actually will work well in patients? Because we would, of course, like to treat as many patients as possible. But it was more basic research question. And it was actually with ISLET that the work I was doing became much more translational. And what do we mean by translational? In my mind, the translational aspect is very clear here. You go from a research protocol where you in the lab can make insulin-producing beta cells from these human embryonic stem cells to something that can be produced in sufficient quantities in a safe way in clean room facilities, because that's necessary if you're going to put the cells into patients. That's quite complex. It's also very expensive, but that is one of the major aims of ISLET. And let me stop there a little bit. Maybe some of you are not completely familiar with cell therapy. So you all know that organ transplant has been around for a very long time. So if one organ in our body fails, if you're able to find a donor that matches you, you can get a new organ, and that can work very well. Now, cell therapy is where you do the same thing. You transplant cells, but it's not a whole organ. And this concept of replacing cells that the patient have lost or that are dysfunctional is routinely used in the clinics today. For instance, when you have skin injuries, like burn injuries, so you can grow skin in vitro and put them back put the skin back on the patient. Bone marrow transplantation is also something which is commonly used. So cell therapy in type 1 diabetes was for some time not known whether it would work, but when the first islet transplantations were done, I think this was very important because it provided proof of principle data that cell therapy actually works in type 1 diabetes, because the patients, when they receive donor islets, they can control their glucose levels in principle without exogenous insulin. Now, we know that the aim is, of course, to treat as many patients as possible. So one would like to have a source of beta cells which provides basically as many cells as you would need, and donor pancreata would not suffice to replace all the necessary cells to all patients who have type 1 diabetes. So this is why the stem cells became of great interest, because what's unique with these human embryonic stem cells, similar cells are also called human iPS cells. What's unique with these stem cells is that they can self-replicate forever, so you can make as many cells as you want, and they have the capacity to become all our different cell types in our body. So we have around 200 different cell types, including insulin-producing beta cells. So if we can devise, manufacture protocols which are very reliable and scalable, we can produce beta cells which are more or less the same every time we make them. And this is, of course, a great advantage. So do we know today that stem cell-derived beta cells are as capable as donor islets to restore normal glycemia in patients? And the answer is yes. So this was extremely important new data that was provided by a biotech company in the US, Vertex, and also a Chinese research group. And they both show that when patients receive stem cell-derived beta cells in sufficient quantities, these patients can become insulin independent. Now, this is very early time, so far, as far as I know, it looks good, but we need to follow these patients for a longer period to know that this treatment is safe. I should note that in one of these trials, the cells were put into the liver, like we do with donor islets. And in the other study, the cells were put above the stomach muscle. So it was very close to the skin. So it will be very interesting to follow also whether the site where you put the cells matters. We need more products, because I think I mentioned to you that the bar will be very high to motivate to transplant these cells into the majority of patients, because for many patients it works absolutely fine with the glucose sensors and insulin pumps. But we also know that even if it works fine, there will never be as precise regulation of the sugar levels throughout the lifetime of a patient as if you had the normal cells. So in Europe, basically what I've been doing is to lead this program to develop new cell products to, in a similar way, move forward towards products that can help patients with type 1 diabetes. So in ISLET, the original goal of ISLET was to, I think in my mind, the primary goal was to develop the first cell product. And at the end of ISLET, we would be in a position where we would be more or less ready to apply for a first-in-human trial with the product that we developed. But since the intention with ISLET is not only to do, to make one product and then the whole thing falls apart, I think we all who worked in this field realized that the first product will never be perfect. We need to continuously evaluate the products in patients and then, improve them as time goes by until we have something which is really good. And I will come back to this because there is also the the aspect of the immunological challenges that we have, which needs to be resolved if we're going to be able to treat all patients with type 1 diabetes. So we also initiated a separate, in parallel with developing the first product, we also agreed on what could be, important steps to move towards a better product. So I think, there were two things we were interested in: to, focus on safety, because so far in these two trials that I mentioned, the product we don't know much about the product, at least with the company, because they haven't disclosed information about the product. But it's pretty clear it's a heterogeneous cell population. So the cells that are transplanted is not 100% insulin-producing beta cells. There are other cells also. So the question will always be, is there a problem with the other cells in the transplant? And that's where the safety comes in. So what everyone has been interested in doing, can we purify the beta cells and show that this is a safer, maybe even a better product? And of course, the obvious challenge is when you purify cells, when you manufacture the cells, you lose a lot of the cells. So the cell yield is a major barrier. But we needed first to start to see, ask the question, is a more pure population more efficient and also safer? So this was one part of the project. We also, in fact, all protocols that have been used to make these cells from stem cells are rather empirical. What I mean with empirical, the protocols have developed through testing a lot of substances, chemicals in the medium, and see what turns the cells into a beta cell. And we really don't understand what these chemicals are doing. So if we understand more about the normal signals that normally does this during fetal development, we can achieve several things. We can achieve a more robust process. What I mean by robust, that it's every time you do it, it works. The other thing is also that we may be able to do it at lower cost. Because in the end, if we make a very good product, it has to be cost effective. Otherwise, we will never get a company to market the product to hospitals and caretakers. So this is something we've been also, one can say this is basic research questions, asking, for instance, when we make these early steps during fetal development towards an insulin-producing beta cells, what is the specific signal that instructs these early immature cells to become a beta cell and not a glucagon-producing alpha cell, for instance? That kind of knowledge has not been around before. And so this has been one part of the, but the focus has always been, can we use this information to make products better? So it's not like we're doing basic research just by curiosity. Then we have also focused a lot on improving quality controls. When we, in the manufacture process, one has to, when we make beta cells from stem cells, that's the time that takes is about a month. And during that process, we have to test at different time intervals whether we are on the right track. What that means is that we do a test and then we can predict, okay, this looks like we're going to make a good beta cell at the end. And those quality controls have been rather, I would say, a little bit old-fashioned and not so quantitative. So there's really a room for improvements here. So this is something we also been working on by implementing new technologies like proteomics, lipidomics, and also single-cell sequencing also. So, and then the last thing I would mention that's been very exciting throughout ISLET has been the outreach activities. And because, I think, first of all, it's very important that everyone who has type 1 diabetes knows about what we're doing, why we are doing it, where we are, what are the next steps. It's also important for stakeholders, university, academic institutions, companies, to understand the complexity of doing it, but also the opportunities, obviously. So if you do all this work in a company, it's very difficult to follow this. And I made a choice very early on that I wanted to do all this work in an academic setting, because I realized that companies today are not, their R&D activities are not suited to solve all these problems that has been facing us. I think the private side is more suitable to come in once we have a product that works in patients, and then it's development work. So this has been my own sort of strategy that has been based on my own experience as a scientist, but also my experience working with industry. I've also started a few companies in this field also, which has been very interesting, and I learned a lot about this. So then what have we achieved? So what's extremely exciting is that this, what we call the first generation product, are now going into preclinical studies. So basically, after ISLET was completed, we managed to finish all the development work of this product. Essentially, we have tested the function of this product in diabetic animal models. We know it's safe, it's very efficient, it's robust, and we have replaced the research reagents with what we called GMP reagents. These are reagents which you have to use in clean facilities before you use the product in patients. So I think this is a very major achievement of ISLET, and I think my hope is that this will be testing this product in patients is going to be, moving this field further in Europe, and hopefully it also will inspire even, hospital scientists and patients outside of Europe. The other aspect I would like to mention is that we have resolved several basic research questions that I mentioned before, which have also now begun to be implemented in new protocols for making improved products that are also can be produced with lower costs. The other thing I mentioned to you, to start thinking about safer and more efficient products. That part has also been very successful, and so part of the team has been able to generate products that are more enriched for beta cells, and that are safer to use in terms of some of the side effects if you have too much contaminating cells in the product. So this is very encouraging. multi-omics approach to improve quality controls has also generated unexpected, interesting data on how we can assess the health of the cells when we manufacture cells using actually the lipid profile of the cells, which was rather unexpected. So this is also exciting. So I think, to just sum up, where we stand today is that we have developed a product now which has been undergoing sufficient tests to move into the preclinical and the clinical pipeline. And there's a lot of spin-off effects of the science that has been carried out, the basic science, and also the new ideas of making more refined products. And I hope we've been spreading all the information, and it's been, I think I forgot to mention that part of the outreach activities, which has been very important, is for the young scientists in ISLET, because they have been primarily involved in this work, not the old farts like me. I've seen on them, I've talked to them, and I know that it's very inspiring for them to talk to, for instance, patients with type 1 diabetes. They feel so much more motivated continuing this important line of work. So I think I stop there, and you're going to hear from some of the other people in the program, and then I'm happy to answer questions.
Thanks so much, Henrik, and thanks so much for that really comprehensive overview. Next, we'd like to invite Professor Elko de Koning, who's the head of the LUMC Diabetes Center and the coordinator for the Human Islet Transplantation Program in the Netherlands, to give us a clinician's perspective. So, Elko, the floor is yours.
Thank you, Cameron, and thank you, Henrik, for describing ISLET. I think what is so important about ISLET is that it's also a way of getting academic centers in Europe together to work on this really important topic of finding real solutions for people living with type 1 diabetes. So in my daily work, I lead a research group on cell transplantation, on islet transplantation. At the same time, I'm an endocrinologist, seeing also people living with type 1 diabetes in my outpatient clinic, and running the islet transplant program. So I'll share a couple of slides I've been asked to illustrate kind of the clinical perspective, and also what should the future entail when we go to solutions with minimal side effects. So in Leiden, we, as Henrik already mentioned, we do organ transplantation, so pancreas transplantation, and we do islet transplantation, and we have a diabetes center focused on people living with type 1 diabetes. So pancreas transplantation has been around for a very long time, but there's two major issues, and that it's a very major operation. And when you get cells from somebody else, you need lifelong immunosuppression, and especially this lifelong immunosuppression. It's not an intelligent drug that only prevents rejection, but it lowers the ability of your immune system to deal with infections and to deal with certain types of, for example, virus-related cancers like skin cancer. So lifelong immunosuppression is really an issue that plays a really important role when you say, well, should I or should I not have a transplantation? And what happens is that the major problems with an organ transplantation, with an entire pancreas, is not due to the islets, but it's due to the tissue that makes enzymes, because 99% of the pancreas makes enzymes. So that's when people started to say, well, we only actually need the islets for people living with type 1 diabetes. We don't need all this tissue that makes enzymes. So that's when islet transplantation started. We've been doing that here in Leiden for the past 20 years, and that is in different centers in Europe, in about 12 centers in Europe. We have active islet transplantation programs. And what happens is that the islets are actually infused into the liver through a blood vessel that runs from the gut to the liver. So the islets get stuck in the liver, and this is where they find a new home actually in the liver. But again, these islets are from organ donors, so lifelong immunosuppression is needed. And then was this paper published last year, and Henrik also mentioned it, when an alternative cell source or embryonic stem cells were used to generate stem cell derived islets. And this was a really important proof of concept because in this paper, we showed that 10 out of 12 people living with type 1 diabetes were with insulin after 1 year. So the people taking part in this trial, and in general, the people who have an indication for islet transplantation, those are people with severe glycemic lability, often characterized by reduced awareness for hypoglycemia and repetitive hypoglycemic episodes when you need help from third-party persons. So it's really a very small group of people. Now, when we talk about regenerative medicine, it's repair and replacement of tissues. Diabetes is at the forefront, together with some other diseases. And why? Actually, because we only need one cell type. It's the insulin-producing cells. Of course, there are also other cell types in the islets, but the key cell type is really the insulin-producing cells. And also other diseases that you may hear about when it comes to stem cell-derived products is Parkinson's disease. You only need one type of brain cells, or a specific type of blindness where you only need one type of retinal cell. So I think that's why diabetes is so in the spotlight when we talk about stem cell-derived products. Now, looking at the future. So if you look at this kind of cartoon, as long as immunosuppression is needed, so that needs to be given for lifelong, so to speak, this treatment will always be for a very small group of people. So whether it is donor pancreas transplantation or donor islet transplantation, or also stem cell islet transplantation, when the cells are derived from cells that are not the same as your own cells and immunosuppression is still needed, it will always be for a small group of people. But of course, there are so many people living with type 1 diabetes in Europe, more than 2 million. We need a solution for everyone. And then there are actually 3 kind of options that could be relevant when we talk about future application of stem cell-derived islet transplantation, because of course we need the stem cells in order to have enough cells for everyone, but without immunosuppression. And I'll just quickly go into the concept of this. Now, what we can nowadays do is basically generate so-called induced pluripotent stem cells from one's own body. So we take some cells, either cells from the skin or cells from the blood, and we can reprogram these cells to make induced pluripotent stem cells. These stem cells we can then mature again to islets. We can do all kinds of quality assessment on these islets, and then potentially we could give these islets back. So these are your own islets. And potentially then no immunosuppression needs to be provided. And indeed, as Henrik mentioned, 2 years ago, there was this publication from China of a person with type 1 diabetes, where they made these stem cells from the own body and then generated islets from them and gave it back to the woman. So she is the first person with the disease to be treated using cells that were extracted from her own body. So again, it's also a proof of concept that this is possible. So the second one is that perhaps we can protect the islets within some kind of protective device. And this is what we call encapsulation. So think of a tea bag or a Senseo pad with coffee in it, and then no tea or coffee, but then stem cell derived islets within this tea bag, where the pores of the membrane are so tiny that immune cells cannot enter. So we could transplant it, for example, under the skin. Immune cells cannot enter, but glucose and insulin can diffuse across the membrane. So also some trials or a trial has been done using this system. Of course, you can also think about it as a shark cage with islets in the cage, but also the integrity of this device, of course, is important, because you don't want if there is a damage or anything, immune cells could enter. However, there was also a trial done that was stopped last year because the functionality of the islets in the device that was made was not very good. So still there is people, especially specific biotechnology companies working on biomaterials, that are looking at this, but it's a little bit lower on the priority list at the moment, I would say. And then there is the third option of how can we make sure that people do not need this immunosuppression in the future, and that is perhaps that we can change these pluripotent stem cells to some extent. And we can do that with a kind of molecular technique. CRISPR is a name that has been around for already some time, where we can make very small DNA changes in the pluripotent stem cell line. Then we can let these stem cells renew, so you have an unlimited source, and then mature them so that they are islets. And in that case, if there are small DNA changes, these immune cells hopefully then would not attack your islets. And this is also a promising kind of avenue that many people in the field pursue. So these are really the kind of potential ways that we say, okay, we can do donor islet pancreatation, we can do donor pancreas transplantation, donor islet transplantation. The first study with stem cell islet transplantation has been done, but still immunosuppression is needed. For the future, we also need, if we want to transplant everyone in the end, go to a product where much lower dose or no immunosuppression is needed. And this is perhaps with the gene editing, you could say it's kind of the invisibility cloak for Harry Potter. And I think the kind of gene editing is probably the most interesting avenue at the moment. Okay, so my last slide is that in Europe we need to come together. That's what we did with ISLET under the leadership of Henrik, to get people together that look at this problem from different angles, clinicians, basic scientists, translational scientists, and to create an infrastructure and to create knowledge in order to go to the next product. And what is really important, that also biotech companies are important, academic institutions are important to kind of lead the way for regenerative medicine and for this new solution. And in the EU, there is this act called the Biotech Act, which basically says how can we lower the hurdles for implementation of these products, for testing it in people living with different diseases, also type 1 diabetes, but also how can we stimulate companies to be part of it? Because as soon as we have a product, we also need companies to pick it up and to upscale. So I think this is also important. So not only the science part, but also getting Europe ready for adopting this new regenerative medicine solutions also for people with type 1 diabetes. And I'd like to finish with this.
Thank you so much, Elko. Really appreciate that overview. So next, I'd like to invite Nicole Rogers, who's a doctoral researcher in the Helmholtz Diabetes Research Center in Munich, to give her experience of being part of the ISLET project and an early career researcher in diabetes. So Nicole, over to you.
Thank you. Elko and Henrik have given a great overview, but I'm going to be talking a bit more about what it was like to work as a youngish doctoral researcher So I'm Nicole, and I work in Professor Heiko Lickert's lab. We're also based in Munich at the Helmholtz Diabetes Center, and I started around 5 years ago. I started my PhD 5 years ago, and so we worked on the ISLET project. So a colleague, Ionikis Etiono, and we worked on trying to improve kind of the stem cell product. So as Henrik briefly mentioned before, we tried to kind of optimize the cellular composition of it by kind of defining what is the best ratio of alpha and beta cells by using antibody markers, and then seeing how well they function compared to other cellular compositions, and really trying to optimize it in the sense of functionality as well as safety. And we also managed to identify like 3 markers to really show, is this safe for transplantation or is it not safe for transplantation? So this we started 5 years ago, and we now managed to publish a little something about that. But actually initially when I started my PhD, I didn't know much about the diabetes field. I was more driven by my passion of stem cells. So I did my master thesis in Copenhagen in a lab working with neurons, and we used stem cells to differentiate them into neurons. And I just fell in love with stem cells and their endless possibilities that they offer. And I joined this project because we were working with stem cells and also trying to help to develop a stem cell replacement therapy, which I thought was amazing and really cool. And it's an amazing field because also, like they both have said, in the past 5 years, I mean, where I was, involved in it, so many things have changed and so many developments have been achieved. It's really incredible. I mean, also these 2 studies that they mentioned, but also, for example, with Parkinson's disease, also the treatment for that. I mean, the field of stem cell or regenerative medicine, it's really incredible, and I'm really happy to be a part of it. But a really nice thing about the ISLET consortium was actually also the fact that, there's like 7 different labs or PIs that were part of it from different countries and cities, and to be a part of that and to meet all of these people. I mean, sometimes you read the papers that they have published and you think, oh my God, they're amazing. And then you get to meet them in person and talk to them and, get all of their knowledge as well. So it was a pretty special place to be in, really. And to also work with the other scientists with the different expertise. And I mean, you could just, learn a lot. So it was great to be a part of it. And, we had annual meetings. So it was also a great opportunity to travel a little bit. So when we had the annual meetings, we also spent a few days together. And so in that regard, it was really nice, and a nice opportunity to also network with other doctoral researchers in different countries. And in that regard, it was really nice. But the ISLET consortium didn't just offer that, but also, as Henrik has also mentioned, these outreach programs. I mean, so we got in contact with Konstantina and with Cameron, who organized the webinar. And it was really a great experience. We did a few things together, and we also bumped into each other at conferences. So that was really nice. But as I said before, I had never really been in contact with the diabetes field that much. And in the lab, you learn a lot about the scientific part. Okay, how does the pancreas develop, and how does glucose regulation work, and all of this kind of stuff. Really, the science part of it, but to actually, it was really nice or good to hear and learn about what it means to be diabetic, and to see what people with lived experience go through on a daily basis, because I wasn't fully aware that it really is a challenge every single day to monitor your blood glucose and to inject insulin every single day and to have constantly needles inside of you. This is not something that you are aware of in the lab. And so it was really good to talk to them and to learn about what it's like. And because at the end of the day, this is, the stem cell replacement therapy, that's, this is for them, and we're trying to, make it for them. And hopefully one day, if they want to, they can have this opportunity to get this replacement therapy. But so they really helped us in understanding and, keeping the big picture in mind for cell replacement therapy and for what we do on a daily basis. It's very easy to get into this rabbit hole, and you're in cell culture all the time, and you get contamination, you're upset, you're frustrated, and then you have to think about the bigger picture, and that helps you keep your sanity a little bit sometimes. So overall, I'm really happy that I was able to join this great project and meet all of these amazing people. And thank you. I think that's it from my side.
Thanks so much, Nicole, and thanks for sharing your experience. The last speaker that we'll have for this session and today is Dr. Konstantina Bumaki, who's a young person living with type 1 diabetes and a diabetes advocate, to give the experience of the lived experience element of this particular work. So, Konstantina, over to you.
Thank you so much. So I have been living with type 1 diabetes for 20 years, and a lot has changed over these years, but diabetes is always there. And as Nicole said also, it is not something you manage once or twice a day. It is there in every decision: what you eat, how you sleep, how you travel, how you work, even how you feel emotionally. And people often see the devices, but they don't always see the thinking behind them, the constant calculations, the alarms during the night, the fear of low or high blood sugar, the frustration when you do everything right and it still doesn't work. And most importantly, the fact that you never really get a break. So there has been real progress. When I was diagnosed, management was much more limited. I used syringes for many years, and I was very little, so I was getting bruised a lot by the syringes. My meter was, taking longer, and I had hypo every night. And I couldn't change that because that was the system that I had. I would just needed to wake up every day at 2 a.m. And today we have technology that has improved safety and the flexibility, closed loop systems, sensors, pumps, and they really have changed daily life for many of us. Like for me, when I joined the closed loop system club, I could sleep through the night for the first time, and this changed my energy throughout the day. This was very important for me. But they haven't removed the burden, of course. You still need the knowledge, the attention, and the mental energy every single day. And not everyone has access to these technologies equally. So this is why something like the cell replacement therapy is so powerful, because for many of us, it represents real hope. The idea that one day we may not have to constantly manage diabetes ourselves, that maybe we could step away from this 24/7 responsibility. That is something very emotional, but at the same time, our community has learned to be cautious. We have heard promises before. When I was diagnosed 20 years ago, I was told by my doctor, in 5 years, we will have a cure, so don't worry. But yet, here we are. Of course, this takes time, and we understand that. So the important factor is the engagement. And I was really happy to join this ISLET program with IDF Europe. And we went to the research center to see them on the field. We have a great communication with many of the PhD students there. And we understood so many things that we could never even Google or search online or even read about them in books because we were there and had a direct contact. And this felt very very important for us, but for them too. We saw, as Nicole said before, she didn't even know how we live with diabetes, and she put a face in this work that she was doing. And this is very important, but for us too, to see what they're doing, this might be a cure for us. And we asked all our questions that might feel stupid, but they weren't. And this was very important for all of us. And so what does the community needs from the scientific and clinical community? I think we need two main things. The first is the clear and honest communication, not over-promising, not too technical, but also not simplifying so much the important details. And second, the meaningful involvement. People with diabetes should be involved in setting research priorities, designing studies, defining outcomes, shaping how results are communicated. And this involvement needs to be diverse because type 1 does not look the same for everyone. And before people fully embrace something like cell therapy, there are some key questions that really matter, that we need to know. What will change in my everyday life? Will this reduce the burden or just change it? What are the risks and long-term effects? Would immunosuppression be needed? Will it be accessible or only for a small group? Will the quality of life be considered, not only the clinical outcomes? Because at the end of the day, success is not just about the glucose level. It is about how we, the patients, live with it.
Thank you so much, Constantina. We really appreciate your experience. We have about 8 or so minutes left for questions. There is a Q&A function down the bottom that people should be able to use. If there's a question that you would like to ask, you can type it in there for the panelists to see. So, Henrik, there's a question here already. Do you believe islet transplantation is essential, given that intact islet function can both upregulate and downregulate blood glucose levels? This means that not only beta cells are required, but also other cellular components within pancreatic islets.
Yes, very good question. I think, to be honest, we don't know yet. I mean, the best way of testing that is, of course, engineer products which only contains beta cells or a mix of alpha and beta cells. So there are, of course, animal studies, but the animal studies are sort of doesn't give a super clear picture. I think that the question is relevant also from the perspective that the patients still have alpha cells left in the pancreas, but whether the beta cell function in the stem cell derived product requires cell interactions with alpha cells, we don't know. I think that the product so far that's been engineered from stem cells contain a mix of hormone producing cells, islet cells. So no one has yet put in patients products that only contains beta cells. And since they seem to work, and that there's a lot of evidence suggesting that they do depend on one another, I would presume that the best product would be a mix of at least beta cells and alpha cells. I'm sure Elko can give his perspective on this also.
Yeah, I agree with what you say. I mean, we don't know, but if we think that nature has done a pretty good job in making islets with a specific purpose, where you have beta cells and alpha cells, probably it's best to mimic nature as well as possible. But of course, we also know now, and as Constantina said, with the hybrid closed loop systems, we only have insulin, and we can achieve already pretty good control despite all the technology burden that there still is with only insulin. But in the end, I think that islets containing all the cell types are probably best.
I don't see any questions in the chat.
There's one more in the Q&A section here that says, "Thanks a lot for the nice talks. My question is with cell therapy, how significant is its potential in treating type 2 diabetes, especially when pancreatic beta cells are severely depleted?"
Yeah, that's also a very good question. And although this feed focus a lot on type 1, there's clearly going to be situations where cell therapy could also benefit patients with that kind of form of type 2 diabetes. And the benefit, I mean, in one way, when we have discussed, the first type of clinical trials, in a way you would like to have, because what we want to do initially is to test the cell's safety and efficacy. The challenge we have today in the first trials that's been carried out is that the patients that are recruited are very, very sick patients, because these are the only ones that we can ethically motivate to use immunosuppression. Now, if you have a patient who has type 2 diabetes with the depletion of beta cells, as you mentioned, and also thinking about what Elko mentioned, that one way to get around the use of immunosuppression is to use your own cells, right? So in these patients, theoretically, you could engineer the beta cells from the patients, and you won't have the problem of autoimmunity. So you could test the cells without immunosuppression in these patients, which would be potentially a situation where the health of the patient is better, and then of course it's always easier with follow-ups and sticking to the clinical program and the routine that the patient should follow. So yes, that's a good point. I'm sure that's going to be useful.
Okay, great. We have one more question. We probably have time for maybe one or two more questions. So this is a great overview of the program, Henrik. Congratulations for the success. I'm curious to know what approaches are you adopting for scaling stem cell-derived islets, full suspension or combined with 2D planar at the early stages? What is your key approach to ensure reliable cell production, ensuring safety from off-target cells? Thank you.
Yes, again, great question. So first of all, no one has solved scalability. I think this is one of the most important challenges to address in the future. We decided that we can't solve everything in the beginning, so we made sure that the cell yield we have in our differentiation, and actually we do use 2D to 3D. So there is significant cell loss when you use that, and all protocols today, both I think Vertex, but also the Chinese group use similar approaches. No one has been able to start with 3D culture from the beginning. We tried that actually for a couple of years, and we could never get it to work. So the cell yield we have is sufficient to do a phase 1 safety trial with around 10, 12 patients. But if you're going to move on, no one has really resolved this. And I don't have a solution to this. I think it's, here we have to work together with engineers, people, CDMOs who have a lot of experience from manufacturing cells. But clearly there's a need for innovation here. And let's say that the cells, the products that's been developed by Vertex in China or even from in our lab, if it turns out the product actually works, and this immune or this shielding the cells from the immune system, if soon there is a solution to this, in theory, you would be in a situation where you would need to make cells for many patients with type 1. And then the major problem will be scalability, because no one has solved this. So yeah, I'm sorry I can't give an answer to that, but we are trying to find new partners to work with who and try to sort of take new avenues to resolve this. Yeah.
Okay, thank you so much, Henrik. I think we're just about at time there, but maybe if we just have one last question to everybody, in 30 seconds or less. What's your biggest hope, or where would you like to see the field in 5 years' time? And maybe Elko, we'll start with yourself.
I hope that we can find a solution for the lifelong immunosuppression part. So if we can have a product that we can transplant when patients do not have to take immunosuppression, I think that would be a major achievement.
Okay, great. Thank you, Elko. Nicole?
Sorry. Yeah. I mean, I think also immunosuppression, but maybe also for, I don't know, everyone or for the society and the public to be open to cell replacement therapy and being, you know, I don't know, more that it's more in touch with people, the stem cell side and their use, and being open to stem cell replacement therapy. Yeah.
Great. Thanks. Konstantina?
My hope would be to have to have it successful, that I could be able to use it in many other patients, of course, and to reduce the burden of diabetes, and to remember how it is to live without this constant thinking.
Thank you. And Henrik, last word to yourself.
So I agree with all of these points. I think that something we haven't touched upon, which is going to be very important, is how we fund this type of work. It's very expensive, and our society has decided that it's up to the companies, the private side, to fund this work. I mean, EU is helping, but this is insufficient. And so I think that we have to put pressure on our politicians to learn more about the complexity of this, and the role of the academia and the role of the private side. And the only way they can learn that is to talk to people who have actually taken this route, who started with the basic research, came up with a product, showed that it works in patients, and it's been used in the clinics today. Because there's not that many people who've done that. You can count them on one hand, globally, actually. And these people sit on very important information that should be utilized. So I think this is important.
Great. Thank you so much, Henrik. Thank you so much, everyone, for joining us on behalf of the ISLET project and IDF Europe. We're really grateful for you participating and taking part and asking some really interesting questions. And to our panelists, thank you so much for not only taking part today, but also the work that you contributed to throughout the ISLET project. And we look forward to working together in the future. So thank you very much, everyone.
Thank you.
Thank you.
Thank you.
Bye.
Thank you.